Yeah, so first of all, I'd like to thank the organizers of the meeting for inviting me. This seems like a really interesting meeting. I've worked in the field of environmental microbiology for a number of years, working in academia, industry, and federal laboratories. And each time I've had an opportunity to work with people from various disciplines. And what's great about that is that everyone brings their own perspective to a project. So you might have one set of data, but you have three different perspectives or five different perspectives, depending on who's there. And usually that works out really, really well. I'll give you some examples later on. So after I retired briefly, I started a small company called Electrochemical. I'm acting as a consultant, and I'm acting as a microbiologist and somewhat of a conduit between electrochemists, microbiologists, biochemists, civil engineers, and chemical engineers. So I'll talk to you a little bit about what we've been up to. From the point of view of electrochemistry and microbiology, you could think of bacteria as small capacitors. Now, in that respect, when they take in food, say glucose, for instance, they also acquire energy in the form of electrons. And electrons are utilized by the cell for power, for energy, and then they're dumped onto terminal electron acceptors like oxygen or nitrate or chromate, R3, so on. There are a number of different terminal electron acceptors bacteria can use. We could use only one, oxygen. So they have some advantages over us, but in a sense, we're like batteries also. So we decided to start looking at bacteria from this point of view. At least I got started, and I think a lot of microbiologists started seeing electrochemistry as a valuable tool because we were trying to understand how bacteria transfer electrons to solid rocks. The reason is there are some bacteria known as dissimilatory metal reducing bacteria. They can use iron minerals or manganese minerals as a terminal electron acceptor. So they could use rocks like you and I use oxygen. And we would measure these interactions by calculating the amount of iron 3 reduced in the form of iron 2. And this is time consuming, but effective. So we decided, I think individually, we all came to the same conclusion that we could substitute an iron oxide with an electrode. And this is an example of the electrodes we've been using primarily through most of this research. It's a flat pattern graphite electrode with counter electrode and working electrode is graphite. Reference electrode is a silver chloride. It's small enough to fit into a serum vial. You can see the serum vial on the right. Let me see if I can. I'll point to it. It's in the serum vial, so it's inside the Viton rubber stopper. So it maintains sterility, it maintains anaerobic conditions, so it's airtight, watertight, and we get really interesting results as far as the electrochemistry. So we combine microbiological studies as well as electrochemistry simultaneously. So I'll show you some of the techniques we've been using that include chronoamperometry, cyclic voltammetry, and electrochemical impedance spectroscopy, which I'll abbreviate to EIS from here on out. With chronoamperometry, basically you poise the potential to a voltage that you want. Quite often we pick the voltage related to particular terminal electron acceptors like you see here. Okay, so so actually let me back up because I wanted to show you. So this is chronoamperometry. So with the chronoamperometry, we started to grow bacteria on electrodes. The bacteria we're using are Shionella, in the genus Shionella. They're really good at transferring electrons to electrodes. So we use this to understand how basically to use the electrochemistry. But when we do that, we had the cells in a suspension or in a solution. Then we added a carbon source. The carbon source was lactate. When we added the lactate, we saw gradually an increase in current. So when we see this increase in current, a microbiologist sees this and thinks, so I have a method to monitor cell growth. An electrochemist sees it and says, this is 1/2 of a fuel cell. And an analytical chemist sees it and says this, calls it a sensor for lactate. So different perspectives are always useful in some of these projects. So next is cyclic voltammetry. Let me know if you disagree. My slide says cyclic voltammetry. And so in this case, we can control the potential and swing it from one direction to another. So if you go from the lower potentials to the higher potentials, we can get oxidation peaks in the presence of a redox couple. And in the opposite direction of the higher potentials to the lower potentials, we get a reduction peak. So when we have a redox couple and it tells us something about the behavior of the reduction peak or reduction and oxidation peaks. So we took the cells and we put them on an electrode and ran cyclic voltammetry. When we did that, we were able to see redox couples associated with, on the far right, a pigment that the bacteria produce, an electroactive pigment known as pyomelanin. And the pigment sorbs to the cell surface. The center couple is a cytochrome, which is an electron transfer mechanism on the cell surface. And the third one is a soluble electron shuttle that the bacteria produce, known as riboflavin. So cyclic voltammetry has been very useful for this. Okay, so as far as electrochemical cleaning, again, let me know if you disagree with what I'm saying, because I think you're seeing the screen that says electrochemical cleaning.
Yeah.
So what we did was the problems with bacteria is that they form biofilms. And when they form biofilms, these biofilms can be very non-productive, especially if you're doing electrochemistry. And we realized that we were running into this problem initially. So after talking to a number of electrochemists, they informed us some techniques that involve electrochemical cleaning of electrodes. And we did just that. We started off with a clean electrode. We got it fouled by just growing Shionella on the surface for a number of weeks. And we were using indium tin oxide because it's a transparent electrode. It allows us to do microscopy as well as electrochemistry. And when we looked at the fouled electrode, the CV, the red CV, is very different than the initial clean CV, which is green. So then after we did electrochemical cleaning, which basically involves sweeping the potential backwards and forwards from -2 to +2 volts, we did this about 3 times at one volt per second. We typically go about 3 times. It may be as many as 10, depending on how fouled the electrode is, and we can tell by the CVs. Then the cleaned electrodes, electrochemically cleaned electrode, is very similar in its CV features than the clean CV. So this is an option that we use every time we run a sample. We always clean the electrode and run CVs to ensure that they're clean. So now you should be seeing a conventional growth study with Clostridium phytofermentans. This is a typical microbial study looking at the growth of this gram-positive organism. It grows anaerobically, and it will grow on cellobios. So we were interested in understanding how it grows in relation to the production of acetate, ethanol, and hydrogen. But at the same time, we also incorporated electrodes to monitor the growth electrochemically. So if you notice, by 40 hours, it's almost completely out of carbon source, carbon and energy source. So when we did our electrochemistry, we saw CVs that didn't really have many features. This is CVA and CVB that correspond to this graph, the second graph, the middle graph. So the arrow for C and the arrow for D correspond to the lower two CVs. Now, the interesting thing about these is that they have a reduction peak around 100 millivolts or so. That reduction peak, if we take the area of that peak, it'll give us the charge density. So in the second graph, we plotted charge density relative to cell growth in terms of optical density. So the red is optical density, it's cell numbers, and the blue is the charge density. So just as the culture ran out of carbon and energy, then it started to pull electrons from the electrode. Now the cells are growing in suspension. So that means that, and they were stirred. So when they came in contact with the electrode, they would just pull electrons out. We were able to correlate electron transfer from the cells as opposed to from the medium, because with cyclic voltammetry, you can also run different scan rates and correlate the scan rate to the peak current, which is on the left. When this correlation is linear, that tells you that charge transfer, the transfer of electrons is occurring at the surface, not in the bulk solution. And when we took the cells out of the solution and just ran CVs of the cell-free medium, we didn't really see any reduction peak. So it indicates to us very strongly that we're having some good luck with understanding how bacteria you can use electrodes for electron uptake. We're using this as we just recently got a patent on this, and we want to try to use this in potentially industrial applications. As an example, if you have a methanogenic consortium for biogas production, this is common in agricultural industries because you have a lot of animal waste, and that can be converted to biomass. I'm sorry, to biogas. But the process is very complicated, and there can be a lot of interruptions to this process, and it can cause the fermenters to malfunction. So what we did is we wanted to see if more than just one species of bacteria can perform this phenomenon. So it's a mixed culture of microorganisms. So when we deprived this mixed culture of any glucose, we saw the charge density increase pretty dramatically. When we gave them glucose as a carbon source, we saw some variability in charge density, but nothing major. We also monitored other things like different chemical production, and ultimately by day 4, we were seeing methane production come up. So this is shown to be a useful and possibly universal tool for monitoring complex interactions like biomass to methane. Okay, so now I'm on electrochemical impedance spectroscopy. And that involves using a sinusoidal AC signal to polarize electrodes and monitor the changes. We look at a range of frequencies from 10 millihertz to 100 kilohertz. We add a small voltage at the working electrode and we measure the resulting current. And what happens is as the cells are polarized, the signal changes, or they change the signal in ways that it causes the phase to shift and the amplitude to decrease. So we can measure impedance. By the way, impedance is a form, it's kind of like resistance, but resistance in an AC signal as opposed to a DC signal. At the working electrode, we read the real impedance. At the counter electrode, we measure the out of phase or imaginary impedance. From these data, we can also derive a number of other parameters. We use software called ZView that calculates all of the parameters based on the equations you see. And one of them is permittivity. Permittivity is a subset of capacitance. So permittivity tells us how easy it is to permit a charge. And that has a lot to do with microbial activity. I'll explain that in the next slide, which is electrochemical impedance spectroscopy in microbiology. So if you have a lot of bacteria, but bacteria have lipid membranes. The lipid membranes are not very conductive. They actually are good insulators. So because of that, it changes the phase. And the greater the phase shift, the more bacteria you'll see. So basically, as bacteria are growing in the solution, you'll see the phase shift get greater and greater relative to cell density. And also, we can look at real permittivity, which is at the working electrode. That tells us something about proton gradients. As cells grow, they have a greater proton gradient. So the more viable the cells are, the more protons they have. If the cells were to lyse, the proton gradient would just disappear and you would see it completely drop. And also there's imaginary admittance, which at the lower frequencies can be used to measure membrane-associated electron flow. At the higher frequencies, it can relate to a cellular biofilm on a surface. So to get more complicated, we can also look at bacteria and define them as equivalent circuits. So with an equivalent circuit, we can take all of our impedance data, run it through a computer, and if we give it the right equivalent circuit as a model, it can model the data based on these circuits, and then we can derive a number of other values. And I'll give you an example. Again, back to our conventional growth study. So here we have the same study we looked at before. Cellobiose is being utilized. And we want to see what's happening with something called the charge transfer resistance. As soon as I find it. Okay, so in this case, we were looking at a Cole plot, which is basically the imaginary or the real impedance, the real permittivity versus the imaginary permittivity. And the distance between the zero and the diameter of the arc, where you see this arc, the larger the arc distance or the radius, the greater the charge transfer resistance. Charge transfer resistance is basically how easy or difficult it is to move electrons. So when you have a new battery, the charge transfer resistance will be very low. But as it runs out of juice, the charge transfer resistance will be very high. If you recharge the battery, the charge transfer resistance will go go back to a very low. Well, this is what we got here too. With bacteria, it's the same thing. So when we looked at this, comparing it to our conventional growth data, the charge transfer resistance is R3 in this equivalent circuit. So R3 tells us that what's going on with the charge transfer resistance. So basically, the cells are beginning to have a difficult time getting electrons about hour 20. Actually, maybe about hour 25. So as the carbon source is being utilized, the charge transfer resistance is going up. It dropped because at 40 hours because they quit growing. And at that time, they're metabolizing because they're spore formers. So as they run out of food, that gives them the signal to go into a dormant state. And in that dormant state, they're making spores. And we think the charge transfer resistance after 40 hours was due to them making spores. And we started off with spores. And then they had to germinate. So what happens is R2 equates to the diffusion of electroactive species. So when spores are germinating, there's a tremendous amount of biophysical activity going on and a lot of ionic flux going across this spore coat. The spore coat has to dissolve, and basically it's sort of like an egg hatching. So the microbe is becoming viable at about 15 hours. When the electrochemist I was working with told us that we had a tremendous amount of activity at around 10 hours, I told them they were crazy because if you look at the microbiology data, there's nothing going on. But then we realized we started with spores. And so I think this is what's going on because R2 equates to the diffusion of electroactive species. R1, by the way, is related to the conductivity of the bulk phase. So that's kind of handy to have because as the bacteria grow and produce acids, that the conductivity might increase, and that's another way we can monitor that. And then we also started talking about how can we monitor a biofilm without contacting the biofilm. Typically, if you want to do electrochemistry on a biofilm, you grow the biofilm on an electrode, and the electrode becomes part of the surface that the biofilm is growing on. We wanted to try to figure out how to monitor a surface without it being part of the experiment. So we grew cells on the surface of an aluminum coupon. So we take the electrode on the upper left. You see the electrode. We turn it parallel to the surface, and we keep inert spacers there to maintain that distance. When we do that, we get results like you see on in the graph. The graph on the upper left is Serratia marcescens. Serratia marcescens is an organism that we encountered that was growing on some aluminum vessels in a national lab, and there's a concern about corrosion of the aluminum, or depletion of the aluminum. So we grew the cells on aluminum and subtracted the sterile controls, and so we get a curve that looks like this. When we looked at the impedance data, we looked at it 3 different ways. One is the sterile coupon. One is the red is the coupon with the biofilm, and then the green is the coupon with the biofilm removed through sonication. So the impedance tells us that the cells were impacting the coupon and they were probably producing acid. In doing so, that increased the conductivity and decreased the resistance. We also looked at admittance, but in the form of imaginary conductivity. That's the sigma double bar. In this way, it allows us to look at admittance that relative to a specific area. So we looked at, again, the peaks gave us the same results without bacteria, either sterile controls or the sonicated. And the peak with the cells gave us a peak that was a little lower, actually significantly lower in frequency. And that's due to the relaxation effect of the bacteria on the surface relative to the impedance signal. And finally, we also saw a drastic increase in permittivity, which is what we expected because the biofilm was an active biofilm. And the increase in the E prime is due to the proton gradient of that biofilm. So as far as future applications, I think we're kind of interested in looking at using some of these techniques in bioreactors relative to pharmaceutical development, bioprocess, monitoring for environmental remediation, either in bioreactors or in the subsurface, as well as agricultural waste to energy, where we can insert electrodes into bioreactors and monitor growth, monitor the bioreactor in real time, and possibly even link that to automated process control. And real-time biofilm analysis is also of interest to us, as well as environmental monitoring. One area we're targeting is red tide or algal blooms. They occur very quickly. It's very difficult to detect them as they happen. So if we have monitors out there in real time, this might be useful. So that's my talk. First of all, I want to acknowledge our collaborating institutions and our funding agencies. Right now, I'm working with South Carolina, University of South Carolina, UNLV, it's University of Las Vegas, Nevada, and the Savannah River National Laboratory. We're developing sensors for pollutant detection. These are biosensors. And I'd also like to mention that new collaborations are welcome. So if you're interested, let me know. Thank you. And I'll stop sharing.